Hi, Mr. Wartsky here. Again, this is the second screencast in nervous system. We end with this slide, and we're going to start with this slide for the second screencast. So the synapse is a space or a junction between two neurons. Now, there are two types. The electric, the neurons actually have a physical connection, and they are connected by gap junctions. And the ions flow between one neuron and the other neuron. And for the life of me, I don't know where any electric synapses are. We're going to focus all of our attention on the chemical synapses, in which there's an actual physical space between two neurons. So the impulse comes down the axon, calcium channels open, calcium enters the axon, and this causes vesicles to fuse with the membrane of the presynaptic bulb. Now the pre means before the synapse bulb, right? So the vesicles will form. I have a picture for you in the next slide. The vesicles open and dump neurotransmitters into the synapse. The neurotransmitters will diffuse across the synapse, bind to the receptors on the postsynaptic dendrite. These are dendrites after the synapse. This will cause ion channels to open, and then when the ion channels open, sodium rushes in, we have depolarization, and off we go down that neuron. Now, enzymes will quickly break down the neurotransmitters and be ready for the next impulse to come down that neuron. Here we go. Calcium enters, the vesicles form, they diffuse, the neurotransmitters go across, they bind to the receptors, sodium rushes in. We have enzymes that will break down these neurotransmitters, and they actually pump right here with this, the neurotransmitter byproducts back into the neuron so they can make some more neurotransmitters. We get all of that. Draw the picture if you like. It's a good picture. Hit pause. Some of the neurotransmitters will cause chloride ion channels to open. This will decrease the charge of the neuron, so it will make it more negative. So it will be harder to reach the threshold. So you will basically inhibit the neuron, and these neurotransmitters are called inhibitory postsynaptic potential, or IPSP. The other neurotransmitters will cause the sodium channels to open. This will increase the charge in the neuron, hit the threshold. It will excite the neuron. These are excitatory postsynaptic potentials, or EPSP, not EPSN or ESPN or ESP or EPS or whatever, but EPSP and IPSP. So what are some neurotransmitters? Well, the more common ones, we have acetylcholine, which is ACH. This is the most common neurotransmitter. It's released at neuromuscular junctions where the neurons attach or excite muscles, and these are EPSPs. Biogenic amines are modified amino acids like epinephrine and norepinephrine, dopamine and serotonin. Talked about those things before. And sometimes they're EPSPs, sometimes they're IPSPs. Now, serotonin is the happy neurotransmitter. Sometimes if you're depressed, you will take this medication that will selectively inhibit the reuptake of serotonin. So serotonin stays in your synapses longer, so hopefully it'll make you happier. These are SSRIs, which are selective serotonin reuptake inhibitors, SSRIs. So they inhibit the reuptake of serotonin. Serotonin stays in the synapse longer. You are happier. Neuropeptides are short amino acid chains. Endorphins are an example of this. Those are IPSPs. They inhibit the firing of certain neurons which are associated with pain and exercise. You can actually be addicted to the endorphins that you produce. They're morphine-like based compounds. Amino acids like GABA, glycine, glutamine, and aspartate, they're usually IPSPs. They inhibit. And those are types of neurotransmitters and their structures. Notice how similar they are. Invertebrates, they have a very simple nervous system. They have a nerve net. Echinoderms have a nerve ring with nerve extending down the arms. They coordinate the arm movement. In bilateral animals, we have something known as cephalization, which is the collection of the nerves in the head. Now, those collection of nerves in the head were initially called ganglion, and the ganglion had a nerve cord associated with it that ran down the body, either on the front or back. The cephalization occurred from flatworms all the way up to arthropods. Echinoderms form a nerve ring, so they're the exception. Now, the ganglion, remember, there's no specialization. It is just a collection of nerves, not really a brain, because brain implies specialization. And here are pictures of the different comparative nervous systems. In vertebrates, and we are vertebrates, we now have two divisions: a central nervous system, which processes all the information, makes sense of the sense, and a peripheral nervous system, which is our input and output. Now, there are two divisions of the peripheral nervous system. We have sensory, which brings all the information to your sensory central nervous system, and the motor division, which allows you to respond to your sense. Now, believe it or not, there are two divisions of the motor division. We have the somatic nervous system, which are attached to your skeletal muscles or your voluntary muscles. You voluntarily move these muscles. Or the autonomic nervous system, and these are smooth and cardiac muscles. These are the muscles that are involuntary. Believe it or not, there are two divisions of the autonomic nervous system. We have the sympathetic, which increases energy usage, and parasympathetic, which decreases energy usage or conserves energy. The central nervous system is composed of the spinal cord and brain. The central nervous system is covered with meninges. These are the membranes. If you have an inflammation of the meninges, you have meningitis. Very dangerous. You know how you can tell if you have meningitis or you don't have meningitis? If you can take your chin and touch it to your chest, you don't have meningitis. If you can't do that, go to the hospital right away. If you have a severe headache, go to the hospital right away. And you can't do that? No, it's not good. So there are three layers. We have the dura mater, which is the outer covering, which is a lot of collagen. The arachnoid matter, it's called that because it looks like a spider web. That's the middle layer, made of collagen and elastin. And the pia mater, which is the innermost layer, that's closest to your nerves and your brain, that's collagen and elastin. Now, the white matter is a collection of myelin sheaths, and the gray matter are the cell bodies. There are the meninges. There's the scalp. That's your skin on top. This is your skull. This is your bone. We have the dura mater. All right, dura mater, the arachnoid matter, and then the pia mater. In the central nervous system, we find spaces or ventricles. These ventricles are filled with cerebrospinal fluid, or CSF. The cerebrospinal fluid is the fluid that's forced out of the capillaries, and it flows from the brain through canals and ventricles down the spinal cord and out into the body where it's absorbed. The central spinal fluid can carry nutrients, hormones, white blood cells. It can also act as a cushion and shock absorber. CSF. The reflex arc is spinal integration, in which the impulse goes from your sensory neuron to your spinal cord, but then goes right back to your motor neuron, and then you respond. That doesn't go to your brain. You don't even think about it. You don't have any idea what's going on, but your spinal cord is making sense of it and sending the response back to your muscles. So here is the circulation of cerebral spinal fluid. Hit pause if you want to read it. Beautiful. Comparative brain. All right. Planarians. There's bilateral symmetry. They have a ganglion with nerve cord. It is the first ganglion because it's the first time we have cephalization. Annelids have two large ganglia that surround the esophagus. The mollusk has the largest and most complex ganglion around the esophagus. Their eyes are amazing. The things that an octopus can do is amazing. Arthropods, they also have 2 ganglia. Brains of vertebrates, we see specialization. Different parts of our brain, they do different things. That's specialization. So we have 3 parts of the brain. We have the forebrain, which is your cerebrum, the midbrain, and hindbrain. Now, the forebrain is where thought and intellect and personality come from. And in a chondrichthyes, if you remove the forebrain, you won't change shark behavior. But a large part of the shark's brain is devoted to smell. They can smell really well. The Osteichthyes brain is more differentiated, and if the forebrain is removed, you change the behavior of the fish. Yes, fish can have personality. I don't believe it, but they tell me that they can. In you people, that's us, 98% of our body's neural tissue is found in the brain, and the average brain is about 1,600 milliliters, 1.6 liters. The male brain is about 10% larger because males are usually larger. The brain is protected by meninges and cerebral spinal fluid. You produce about 500 mils of that per day. There's also the blood-brain barrier, in which astrocytes will surround the capillaries of the blood vessels in the brain to prevent things from moving in and out. Our brain is dominated by the cerebrum, which is the forebrain, and that is divided into 2 hemispheres and covered by the neural cortex, which is a layer of gray matter. Each hemisphere contains ridges, which is a gyrus or gyri, shallow depressions, sucklus or suckli, or deep grooves, a fissure or fissures. The more that you have, the more surface area to volume ratio you have. So the more cells you have, maybe the smarter you are. Babies have smooth brains. As you get older, your brain increases in size. It can't grow anymore, so it just folds in on itself. The forebrain is the seat of higher mental functions, thoughts, sensations, intellect, complex movement. The cerebellum in the back, the second largest part of the brain, has hemispheres, but you just can't see them. It adjusts movement and allows you to do the same movement over and over and over again, something known as muscle memory. The cerebellum is responsible for balance, hand-eye coordination, and muscle memory. Hey, I said that just recently. I know, but I had to say it again. And we have the cerebellum down here. The cerebrum's here. We have all the suckli, fissures, and gyri right there. Yep. And this right here is the spinal cord down there. The medulla oblongata is part of the brainstem. Pons hold everything together, like the structure that holds it all together. You remove the cerebrum and cerebellum, you have the diencephalon. This is made up of the right and left thalamus. And now the thalamus is important. It is the relay and processing center of the sensory information. So everything you sense has to go to a different part of the brain. It goes through the thalamus. So all neurons lead to the thalamus. You'll also find the hypothalamus, and you know that is the center for homeostasis. And the pituitary is also found there. Look at that. It's amazing. Now, if we had more time, we could talk like the corpus callosum right there binds the 2 hemispheres together. There's the thalamus is right here, your relay center. Okay, the midbrain hypothalamus is here, pituitary is right here, the pineal is right here. All right, the pons we said connects everything. The diencephalon, I don't want to do that. Diencephalon is this area right here. All right, we're going to stop there. If you have any questions, please refer to your textbook, or you can listen to the video over, screencast over again, or ask me tomorrow in class. Have a good night.